Locusts have a pretty bad reputation, known for wreaking havoc all over the world. But locusts are also intriguing and complex creatures, a special species, worthy of intense research by scientists around the world.
Locusts have been around as long as humans have been around. They're recorded in the Bible, in the Quran, in Chinese history. Locusts, grasshoppers, belong in the insect order Orthoptera.
The locust outbreaks can be extreme, and wherever they land, they can cause 80 to 100% crop loss.
Locusts threaten people's livestock and food.
They cover a huge extent of landscapes, including different ecosystems and biomes. They cross boundaries, they cover a lot of different cultures, a lot of different legal systems, and so managing them is really challenging.
In 2019, there was a locust outbreak. The outbreak started in Ethiopia and then it migrated to Kenya and then Uganda. When the locust attacked, the food prices went high and then so life became tough for everybody.
Children born during plague years in villages impacted by locusts are much less likely to even start school. So you have long-term impacts on educational outcomes. So locusts can be devastating for livelihoods, but they are also very fascinating from a biological perspective. They have a special phenotypic plasticity. They can transform based on their environment, and they have this sort of Dr. Jekyll, Mr. Hyde switch.
The fascinating thing about a locust is it's just a special grasshopper with a secret ability. A lot of times we think of a gene that might make somebody tall, or like some genes that might code for someone's eye color. But locusts, they have genes which allow them to change into different morphs during their lifetime, depending on the environmental conditions and whether they're right for them. So they just look and act like a normal grasshopper, and then when the right conditions come, sporadic rain or heavy rainfall in normally dry areas, they respond dramatically, and they come out in larger numbers. They sense one another's presence, and that changes their neurochemistry, the color of their bodies. They become attracted to one another, and if these conditions persist, then they march in coordinated formations across the landscape and then eventually swarm in unthinkably large clouds.
This is a textbook example of phenotypic plasticity. Density affects the phenotype. Same species, but reared in two different density conditions. There's a locust, gregarious, and solitary. The only way you can achieve this coloration and then gregarious behavior is by rearing them in high density condition. I have been studying locusts for my entire career, and here I have the luxury of rearing them and can do some very exciting experiments. These things will actually march in the lab. If you have a high enough density, you can actually see them march around.
It's complicated to predict locust outbreaks, which have these horrible impacts on folks in locust endemic regions, because once we think we figured something out, then we put that into our prediction models, and then it doesn't always play out in nature the way that we thought it would based on our lab data.
So when you see an upsurge on the news, there's the tendency to think maybe there could be a simplified solution or we just need a silver bullet, but it's actually highly complex.
For other pests, you can manage them like a country or a community can just try and work on it and then they eliminate the problem. But locusts require collaboration, communication between many people across countries, across communities.
In Mexico, we have a Central American locust. The closest swarm of the Central American locust to the US border is in Tamaulipas. It is important for us to have a steady resources to study. Then when it's time for an outbreak, we need to have a system in place to deal with these things. And we should do that globally, but we should also do that in the US. We now have the capacity to study them in a very systematic way, in a comparative way.
The BPRI is made up of PIs, so principal investigators or researchers at 6 different institutions across the United States, as well as trainees, so students and postdocs that are associated with those labs, all working to tackle this complicated problem that is locusts.
Within the BPRI, we have a remarkable opportunity to study, for example, within species across levels of organization from molecular to the genome on up to their field ecology and what happens in outbreaks. We can conduct the type of comparative analyses that really haven't been possible to this point.
Locusts are actually really great to research organism or study organism to study physiology in the lab. So most of the people who have studied locusts in the past 30 years or so were lab-based scientists. But now we are talking about studying locusts in the field, studying locusts in a context where those actually matter to people. And that's novel.
It's easy to design an integrative research program on paper. We have learned over time, of course, that implementing that concept can come with many challenges, and we've worked to overcome those.
Being in BPRI gives me the opportunity to be able to interact with other people, know how they think. We are all trying to solve the same problem, but we also think differently. So we have these tools that help us to see the problem from different perspectives and therefore able to come up with something that is more complete.
By studying locusts from all of these different angles, we hope to understand not only how we can help people globally that are directly impacted by locusts, but also understand why animals in general are able to shift and change and what kind of parameters are on them.
What's interesting about locust is that this transition from a shy grasshopper to a swarming locust is dramatic. It's anatomical, it's behavioral, it's physiological, it's nutritional, and it's a big change in the animal that happens somewhat rapidly. Trying to understand that requires that you look at all of these coordinating phenomena together. And so you have to have expertise in different aspects of the biology of the animal and technology to be able to come to a good answer.
Of course, one person cannot know everything, but if we bring in people with different expertise at different scales within biological organization, we can actually bring these brain powers together to understand something greater that no single lab or no single scientist can understand. At Behavioral Plasticity Research Institute, we are trying to do that.
This NSF grant is allowing us to move tools into the locust system that have never been in the locust system before. I'm really interested in understanding how bacteria influence their hosts, and specifically I study insect gut bacteria. And because locusts do so many interesting ecological and evolutionary things, I'm interested in how microbes have a role to play there.
So my area of expertise is nutritional physiology and ecology. In collaboration with my students, in particular Chris Brennan, is understanding how nutritional inputs influence gene expression. When it comes to feeding and how they regulate their nutritional inputs, one of the critical resources that the BPRI has developed that hadn't existed previously were high-quality genomes.
The genome is the foundation right now to study any organism. The first thing that happened with BPRI is to put the genomes together and then to help annotate them, and it's now exciting to see how these annotations are being used. The experiments that are done across the institute are mapped and interpreted with the help of these genomes.
It's one of the things that seems most exciting to me.
Erez Lieberman-Aden is one of the lead people sort of in the field of 3D genome sequencing. He's one of the inventors of this technology called Hi-C. Originally, it was put forward to figure out how the human genome folds in 3D. It helps assemble the genomes cheaply, which comes in handy if you need to put together a lot of genomes from different locusts and grasshoppers.
A key application of 3D genomics is its use in the so-called problem of assembling genomes. This is what the Human Genome Project did. It took decades. It cost about $4 billion to do. 3D genomics methods actually bring down the cost very, very, very considerably and make it possible to greatly improve the quality of the results.
In order to be able to analyze a gene of interest or a gene involved in a process of interest and manipulate it to see if it has an effect, you need to be able to manipulate the genome. And so that's where we are trying to transfer the methodologies that work really well in Sophila melanogaster into the locust species. In order to really specifically affect a gene of interest, you need more precise methods. And that's where CRISPR came in. We just used Cas9 protein, which is what cuts the DNA in the presence of a guide that guides it to that particular spot where it should cut. And we in vitro transcribed the guide, and we brought the protein with the guide together, and then injected that with a repair template to bring something in the genome so that we can recognize that we actually made a genome edit. And so that method works at low frequency in other fly species that we tried first before we jumped to locust. And so we're tinkering with these elements, trying to clone them out and bring them into our plasmid sequences to then bring into eggs.
My lab has been working on collision avoidance for more than 20 years. We're interested in how animals perceive objects that are approaching on a collision course and how they generate escape behaviors in response to those. My background is in mathematical physics. I'm interested also in modeling biological systems using mathematical tools. So I do a lot of that also at the level of single cells and how they communicate with each other. One of the reasons why locusts are very interesting for collision avoidance behaviors is that you have the system where we understand the pathway the best. You have actually one single neuron on each side of the brain, one for each eye, that actually is able to respond very selectively to objects that are approaching on the collision course, much less to, let's say, an object that's translating or missing the animal or some other visual stimulus.
What we can do is engineer electrodes to implant in the brain and then record from the brain. It's kind of like a little microphone, and we're listening in on the conversations of different parts of the brain talking to each other. And we can do that while we're presenting olfactory stimuli, so like smells. By doing this, we're hoping to better understand the language by which different neurons talk to each other with. On a genetic level, in order to enact a lot of changes in the brain, there has to be genetic, transcriptomic, epigenetic changes that we haven't even started to investigate.
So once you know what are the genes and you can kind of study them, you want to know how they're expressed in cells. There's a team of people that was doing what we call transcriptomic, which is looking at how genes are expressed either across different phases or within different cell types within the animals, and also across the different phases.
Locust is a very interesting system because of the plasticity. But how to understand it at a single cell level, it is totally unknown. It's triggered by one single cell or triggered by a set of single cells, for instance, in the brain or in the other part of the bodies. So those are the kind of things we want to really decipher. In the lab, what we do is really isolate the single cells, for instance, single neurons from the grasshopper and put it into nanoliter droplet and profile them at a large scale. So we provide those important descriptions for other biologists in the BPRI to study. So we kind of like mapping out the terrain of the Earth, and then other people will look for the interesting mountains to show this is what matters.
You can take the idea from the genetics and try to test it with behavior, test it with physiology. Vivian Peralta Santana, a PhD student in my lab, and she's also a National Science Foundation graduate research fellow. She's interested in understanding the transgenerational phenotypic plasticity. What mother experiences actually may have an important implication for the baby. We know all that, but there's really not a good model system to study. Turns out that locusts are a great model system for studying that. Dr. Maiva Pescher, she's a postdoc at BPRI, and she is working on many things, but her primary focus is to understand the gene expression differences during the change from Solitarius to gregarious transition.
So here we have a beautiful marked female, and I'm going to dissect it immediately to obtain tissue for RNA.
So we certainly hope that we can capitalize on all the tools and all the information that we've gathered in the past 4.5 years to understand much better and in much more detail phase change across different perspectives in the next 5 years, if we are able to continue working on this as a coherent group, which is what we hope.
Any really modern scientific technology, it's the result of human collective action. And that is really true at all levels, right? That's true at the level of scientific collaboration. It's also true at the level of societies that decide to invest in making it possible for people to live stable lives, doing research, who invest in things like the National Science Foundation, and say, hey, this is going to be a part of life in our society. And that choice, I think, makes us all active participants in the scientific destiny of this country and this species.
I grew up in a small village in Pakistan. When I was a kid, seeing the locusts descending upon our fields, farmers were with their pans and pots, whatever they had in their hands, banging them to make sure that the locust goes away from their farm.
They will go into what we call recessions for long periods of time. Environmental conditions will change, there'll be a lot of rainfall. All of a sudden, we have an upsurge and an outbreak. It's this emergency again, where there's been loss of resources, loss of infrastructure, people in charge have changed, folks have retired, the problem has kind of gone away, and then it's back in many countries that have less access to resources and less infrastructure that gets maintained over time.
Anytime there's a significant outbreak, we see that the governments, especially the one in Pakistan, is going to go ahead and cut down the resources for women's education, for women's professional development. Now you have generation of women that are not going to be educated. I mean, that is just mind-blowing, and it is extremely heartbreaking.
The actual management of locusts is just as much a cultural and an economic and a social problem as it is a biological problem. And so there's no way to make this more sustainable without broad collaboration from a lot of stakeholders and disciplines.
This is a global problem, and it requires a global approach. You need people from all over the world in different disciplines: sustainability measures, policymakers, farmers, builders.
At BPRI, we have stakeholder meetings where the scientists bring in the locust control people in the field from different countries. We've done it for Mexico, we've done it for Argentina. The idea is for us to listen.
I joined the Global Locust Initiative Lab at Arizona State University, primarily interested in understanding how I can use my knowledge of biochemistry and apply it to curbing locust outbreaks.
When it comes to data,
Our main goal is really to engage key actors from locust management and research to forge sustainable solutions and partnerships around the globe. Our online platform that we call HopperLink, we have 200 members from over 40 different countries. We can come together as a community to share news, updates, and share research.
Many times when scientists study something, those things get published in scientific literature and stay there. We understand their biology really, really well. But what does that mean for people who are affected by locusts in Africa? At BPRI, we're trying to change that in a way that we want to translate our research into something that can actually help people.
This is really exciting because it's so interdisciplinary. We are looking at this problem from genetics and from ecosystems, from molecules to management, if you will. There's this sense that we're really kind of looking at it holistically.
One of the things that this reveals is that there are ramifications from human land management and land tenure that could have impacts on the frequency and duration of these outbreaks in some species. For instance, overgrazing or under-fertilizing land is producing this perfect locust food in some contexts. In Senegal, we looked at this on a broad scale with 100 collaborating farmers.
We have to do a great job also trying to see how different ecosystems will respond to climate change.
We have to work together to solve this challenge, to understand their biology, to understand how we can sustainably manage them. And it's inspiring to talk to people in all these different cultures, different languages all over the world and hear their story, hear their interactions of locusts, how it impacted them growing up, why they're interested in studying them.
We are on the verge of achieving something great, something amazing, something we can push ourselves a bit more. That's not only a scientific achievement, but also can help people. The work shouldn't stop. The problem that we're tackling is much bigger than a 5-year project.
What I would hope for the future is that we can move on this road towards integration and iteration and exchange of ideas among biologists of all different disciplines, among social scientists, among folks from the humanities, and very importantly, stakeholders on the ground who are impacted by locusts.